Good morning everyone, welcome back to my channel. In today's class, we will discuss glycolysis, and it's a very important topic that occurs in every cells of the body, and it also occurs in presence and absence of oxygen in both condition. Okay, so in aerobic condition, that means when it occurs in presence of oxygen, it provides pyruvate, and when it occurs in absence of oxygen, that is the anaerobic condition, it gives us lactate. Okay, so that is why when lactate generate in our body, specifically in muscles, it get flaccid. So glycolysis occurs in two phases, like preparatory phase and payoff phase. Preparatory phase is the preparation of the phase when you have to break the energy, like for your preparation of study, you have to pay fees, right? But when you have completed your study and you complete your study and you are trying for job and you get job, that time that job give you the payment, right? So that is the payoff phase, when you don't have to pay. So why glycolysis is important? Because it's the most important energy generating phase. That's why it is very important to happen. Okay, so in books there are 10 steps, but you may feel that these steps are very much complicated, but these steps are very easy. Okay, basically we need to know the structure of glucose and fructose here. So we all know that these two compounds are 6 carbon compounds and they known as hexose. Okay, so let's start. Firstly, I have written all the steps because to save the time, of course. So firstly, see, this is the structure of glucose, where we can see the first carbon, which is composed of a aldehyde group, that is 8C double bond O group. It is a aldehyde, right? Next you can see the whole glucose formula, HOH, second carbon, third carbon is OHH, fourth is HOH, fifth HOH, and sixth CH2OH. So what happens in the next step, that is, one enzyme works here that convert glucose to glucose 6-phosphate. So what happens? There addition of one phosphate group, that is, see, this is another structure of glucose where only change is in 6 carbon compound, that is the phosphate group addition. And this enzyme named as hexokinase or glucokinase, these two can work at the same time, right? Hexokinase, importance of hexokinase and glucokinase for one step. Why there is two enzyme, right? Because hexokinase can convert this conversion in the low concentration of glucose, when very less amount of glucose is present within our body. Glucokinase can convert this less amount to glucose 6-phosphate. That is why, but glucokinase only works at higher concentration of glucose. That is why these two enzymes work for the same step. Okay, and there is, I have said that energy expenditure. So ATP is splitted into ADP and PI, and there is another cofactor, that is Mg2+. So the next step, what we get glucose 6-phosphate, only for addition of phosphate group in the glucose structure, otherwise the whole structure will remain same. So this phase is known as phase 1. Next phase, these two phase. Okay, so from glucose 6-phosphate there is fructose 6-phosphate. Why fructose? Because for glucose the first carbon is aldehyde, and for fructose the second carbon is a ketone group. Okay, C double bond O. So that is why it's a ketone, and almost every carbon is same, will remain same, and so that this is named as fructose 6-phosphate, because in the 6th carbon only addition of the phosphate group. Okay, and we all know that glucose and fructose are isomer to each other. That is why this step is known as isomerism, and that is why there is a reversible arrow, that from fructose 6-phosphate it can change to glucose 6-phosphate, and glucose 6-phosphate can change to fructose 6-phosphate, and the name of the enzyme is phosphohexoisomerase. Phospho because phosphate group attached, that is why phospho. Hexose because it's a 6 carbon compound, so hexose. And this is a isomerism, so the enzyme named as phosphohexose isomerase. It's very easy. Let's move to the next, that is the phase 3. Here we see at the first carbon we added one phosphate group. This phosphate group is added to the first carbon and as well as in the 6th carbon of a fructose structure. So this is a fructose because its second carbon is the C double bond O, that is a ketone group, so it is a fructose. So firstly it was fructose 6 phosphate and we add one phosphate group to its first carbon. So it's changed to fructose 6 phosphate to now phosphate group is also in the first carbon. So that is why it will named as fructose. Phosphate group is attached to first carbon and the 6th carbon. That is why 1,6-bisphosphate. Fructose 1,6-bisphosphate because phosphate group attached to its first carbon and 6th carbon. So there must be a change. It is not an isomerism. That is why it also need energy expenditure. So ATP converted to ADP plus Pi. And we need the cofactor Mg2 plus. And name of the enzyme phospho because phosphate group is attached. So phospho. Fructo because it's the substrate is fructose. So it's phospho fructo and the activity of the enzyme is kinase type. Okay, so that is why it is named as phosphofructokinase 1. So again, firstly it was glucose. From glucose to glucose 6-phosphate, the enzyme is hexokinase or glucokinase because hexokinase work in high both in low glucose concentration, but glucokinase only get activated in high concentration of glucose. So that is why hexokinase and glucokinase, it's convert glucose to glucose 6-phosphate. Phosphate group addition at its 6th carbon and there is change of ATP to ADP plus Pi and we need Mg2 plus. In the next step, that is the second step, where glucose 6-phosphate is converted to fructose 6-phosphate, it's isomer, and this step is also isomerism. That is why reversible arrow is given here. And this enzyme is named as phosphohexose isomerase because they are 6 carbon compound, that is why hexose, and this is isomerism, so phosphohexo isomerase. Okay, next, from fructose 6-phosphate it's changed to fructose 1,6-bisphosphate. Okay, now there happens a very important thing here in the 4th step. Okay, so this is a 4th step. What happens in the 4th step? That is, from fructose 1,6-bisphosphate, we will cleave this compound into 2,3 carbon compounds. So we will split fructose 1,6-bisphosphate into 3 carbon compounds. Okay, so from where we will split it? From the 4th carbon, right? So this is the first carbon, it's the second, it's the third, and we will split it from here. And we will write this 3 carbon here and other 3 carbon, like this one is here. Okay. So 1, 2, 3, see here is the same CH2OP, that is the first carbon, phosphate group attached, and the second carbon is the what is it? It's a ketone group, so C double bond O, and there is CH2OH, one H is extra because we are cleaving this bond here, so there must be addition of hydrogen to this carbon, so that is why CH2OH. Okay, so this carbon compound is a 3 carbon compound and it has similarity with acetone. So this compound will be named as dihydroxyacetone phosphate. So it will be named as dihydroxyacetone phosphate. Dihydroxyacetone phosphate. Okay, so and the other compound, we cleave that fructose 1,6-bisphosphate into two 3 carbon compounds. So first 3 carbon compound is dihydroxyacetone phosphate, and the second 3 carbon is the glyceraldehyde 3 phosphate. This carbon is the fourth carbon here. So HC double bond O, it will become an aldehyde because we are cleaving this, breaking this bond here. So we will get one hydrogen attached to this carbon, and this group will change to HC double bond O. It's a aldehyde. HOH and CH2OP, this 6 carbon will remain same. So phosphate group attached to its third carbon position, that is why, and this structure has the similarity with glycerol. So this is named as glycerol with aldehyde and phosphate. That is why this compound is named as glyceraldehyde 3 phosphate. Glyceraldehyde 3 phosphate. Okay. Next, because glycerol formula key, glycerol is CH2OH, CHOH, CH2OH, and this glyceraldehyde 3 phosphate is similar with glycerol, only its first carbon is aldehyde, that is why it is glyceraldehyde, and it has also phosphate, that is why glyceraldehyde 3 phosphate. Okay, so once again there is a change. What change? That there is fructose 1,6 bisphosphate, and we split it into two 3 carbon compound. First one is the dihydroxyacetone phosphate, and the second one is the glyceraldehyde 3 phosphate, and these two are isomer because they have the same structural formula. Okay, so that is why they are also isomer. Who are isomer? This dihydroxyacetone phosphate and the glyceraldehyde 3 phosphate. That is why here the isomerism, there is reversible arrow is given here, and the enzyme, name of the enzyme is phosphotriose isomerase, because it's a 3 carbon compound, that is why triose, and phosphate group attached to both of them, that is why phosphotriose, and the property is isomerism. So phosphotriose isomerism, or isomerase is the enzyme. Okay, in the next part, so that is the fourth part. Fourth stage is to cleave this fructose 1,6 bisphosphate into these two 3 carbon compounds, and these two 3 carbon compounds can change to their form, like dihydroxyacetone phosphate is also changed to glyceraldehyde 3 phosphate, that is why they are isomer. So this is the fifth step. Okay, so fifth step is also a isomerism. And why we are taking glyceraldehyde 3 phosphate? Because from dihydroxyacetone phosphate we cannot generate energy. For plant we can use dihydroxyacetone phosphate, but for humans we cannot generate energy from dihydroxyacetone phosphate. That is why we are converting dihydroxyacetone phosphate to its functional form, that is the glyceraldehyde 3 phosphate. Now step is the sixth step. So from the sixth step we start gaining energy, right? We start providing energy. 1 to 5, that is the preparatory phase, where there is no net gain of energy, only energy expenditure happen. But from the sixth phase we start to gain ATP. So we know that the NAD is also a form of ATP. From NAD we get NADH plus H plus, and this is a form of NADH equals to 3 ATP, right? So in this step we will generate 3 ATP in one step. Okay, so from glyceraldehyde 3 phosphate we change it simply here. Glyceraldehyde 3 phosphate, glyceraldehyde 3 phosphate is look like HC double bond O, HOH, CH2OP. So we will convert only this aldehyde group, okay, into carboxylic group. Like carboxylic group is COOH. So that is why we have written this here, COOH. In place of H we add a phosphate group, so that is the COOP. COOP. Okay, this is a carboxylic group, COOH. In place of H we add phosphate, and in the third carbon phosphate added previously, that is why. So with glyceraldehyde 3-phosphate, the modification is 1 phosphate, 1 extra phosphate added to its first carbon. That is why it is named as 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate, and the enzyme is named as, it acts on the glyceraldehyde. Okay, glyceraldehyde. So the enzyme is named as glyceraldehyde 3-phosphate dehydrogenase. Glyceraldehyde 3-phosphate dehydrogenase. And for in that step we generate NADH plus H plus, which is equal to generation of 3 ATP. Okay, so in the next step, so this is the 6th step where we convert glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate. Okay. 1,3-bisphosphoglycerate. This is. From 1,3-bisphosphoglycerate, we will generate, see what is the change? It's very easy. From COOP to COO minus. Simply we omit that phosphate group. So here is the, we remove this phosphate group. Okay, next. So it will become COO minus. It's free from phosphate. There is no hydrogen. So COOH will convert to COO minus. And the second carbon will remain same, HOH. Third carbon is CH2OP. It is also same as previous. So this compound is named as phosphate group added to its third carbon. That is by 3-phosphoglycerate. So it is 3-phosphoglycerate. In the next step, and this is that 1,3-bisphosphoglycerate to 3-phosphoglycerate, the enzyme is named as phosphoglycerate kinase. Okay, and this there occurs also generation of ATP from ADP to ATP and cofactor Mg2+. But this arrow is in 2 direction. So that means this step is also reversible step. So we can convert 3-phosphoglycerate to 1,3-bisphosphoglycerate. They can interchange their form. That is why, so the enzyme named is phosphoglycerate kinase. Okay, so now we generate, so this is the 7th step. In 7th step we also generate energy, ATP. Okay, in 6th stage we generate 3 ATP, and 7th step we generate 1 ATP. Okay, in the 8th stage, 8, from 3-phosphoglycerate, we only change the position of the phosphate group from 3 to 2. See this structure. Everything is same as 3-phosphoglycerate. Only this phosphate group changed to its second carbon. From third carbon to second carbon. That is why this compound is named as 2-phosphoglycerate. From 3-phosphoglycerate to 2-phosphoglycerate. That is also a isomerism. Right, so that is why, and the enzyme who is responsible for changing the position of functional group, because phosphate is a functional group, so the enzyme which is responsible for transfer of or changing the position of a functional group is known as mutase. This is a very important thing. So that is why this is named as phosphoglycerate mutase. So mutase and kinase, you understand that difference? Mutase is only responsible for the transferring of the functional group, and the kinase is responsible for the kinase activity like addition of phosphate group. Okay, so that is the 8th step. 8th step where we do not get any energy, as because this step is simple isomerism. Okay, so sun is here on me. And see, from 2-phosphoglycerate to phosphoenol pyruvate. Now we add enol group. And enol means we need to transfer single bond to double bond. That is why we will found here a double bond. So what happened? One H2O will remove one. See this 2 compound. This is 2-phosphoglycerate, and that is phosphoenol pyruvate. Phosphate group attached to its second carbon as it already added in the previous step, right? But what happens here? that only H2O removed. So how we remove H2O? Like OH from here and H from this carbon. So H2O removed, and this will be COO minus first carbon, second carbon COP, and that is why there is a double bond CH2, and this compound is named as phosphoenol pyruvate, and the enzyme is named as enolase. So enolase will transfer or convert 2-phosphoglycerate to phosphoenol pyruvate by removing one H2O. Okay. So from phosphoenol pyruvate, so this is step 9. See, we almost reached at the 10th step. That is, from phosphoenol pyruvate we will lead to pyruvate. So what we have to do, that we have to remove this phosphate group. So in the next step that phosphate group will be removed. So it will be COO minus, COH and C double bond CH2. That is the pyruvate. But pyruvate in its enol form, as this double bond is still present here. So if you want to make it in keto form, that is also possible. So here is the keto form from the enol form, that is the COO minus, C double bond O, that is a ketone group, and double bond CH3. So that will be the pyruvate keto form. And the enzyme is pyruvate kinase. And this step is irreversible. That is why I have given irreversible arrow here. Which step? This 9th step. From phosphoenol pyruvate, this 10th step, this phosphoenol pyruvate to pyruvate. Phosphoenol pyruvate to pyruvate, the enzyme is pyruvate kinase, and there is also generation of ATP, right? And this cofactor is Mg2+. So here is 2 form available, right? Any reaction is spontaneous, right? So if you leave that reaction for few seconds, it will change into a new compound. So pyruvate enol will be not remain same for the long time. So if you stay that reaction happen for few times, so it will change to its keto form, that is COO minus, C double bond O, CH3. Okay, so it's very easy. And what I have said previously, the glycolysis can occur in presence and absence of oxygen. So in presence of oxygen we get pyruvate, right? But in absence of oxygen we get lactate. So if there is no oxygen, what will happen? That pyruvate, its ketone form, will change to the lactate. So what will happen? The enzyme will be lactate dehydrogenase, and the production of NADH plus H plus to Na plus. Okay. So what happened? That there will be generation of NAD plus to NADH plus H plus. Again 3 ATP will generate in this step, and the enzyme is lactate dehydrogenase, and the compound is COO minus, HOH and CH3. That is lactate. Okay. So this is the whole story. Hope you understand this very well, because I try my best to make you understand this complex topic into its simplest version. And this is very important topic for all kind of students who are preparing for competitive exams also, like CSIR-NET, GATE, and various kind of biotechnology exams. Okay, so every kind of exam you will get any one of question from glycolysis. Okay, so prepare these steps very well, because these are very easy. In one look it looks complicated, but it's very simple. Okay, so stay tuned with me. Hope this video will be very helpful for your preparation. Bye bye for now.